Hi everyone. Tonight we have another talk. Good night, good evening, good afternoon. Let me just start. Okay, this is the name of the talk. Protoplast or spheroplast formation. This was actually the first experiment that we did in this class. It's rather a simple experiment. However, it has high significance because it is explaining the formation of the cell wall, and everybody knows the cell membrane, but cell wall is a different thing in bacteria. Gram positive, gram negative bacteria. Same thing for fungi. They have cell wall as well. Plants, plant cells have cell wall. Animal cells do not, for some reason. So let's start because it's important. Today is the anniversary of the devastating earthquake that happened in Turkey last year. So that's why I wanted to do this talk tonight because it's about walls and protection and life. So let me start. It's rather short, shorter than the other ones, but let's see. Hopefully I can finish it in 45 minutes or an hour. So, introduction. Gram-negative bacteria membranes are composed of several compartments. So if you are into microbiology, you know there are gram-positive bacteria and gram-negative bacteria, and there are huge differences in between them. Their especially outer membranes are structured differently. That's why if they are stained with the gram, that's where the name is coming from. Gram stain. If they are stained positive, meaning they have a thicker cell wall, they are called gram positive. They have a bigger, thicker cell wall. The material, cell wall material is thicker. However, the gram-negative bacteria does not have such cell wall. So that's why they're kind of one is positive, one is negative in terms of being stained with this material, dye, gram. Whoever found it, I guess the person found it, named it after himself, herself. So gram-negative bacteria membranes are composed of several compartments, respectively cell membrane, peptidoglycan cell wall, periplasm, and outer membrane. I wish I had a drawing here, but even those days, it was just because most of the labs were studying bacteria, and we were studying yeast and there are huge differences, between, even though they're all cellular units and cellular structures, but the way their membranes and their cell walls are structured, constructed, they are all different. So depending on the lab, and like I said, most of the labs were studying bacteria. We were the only kind of eukaryotic yeast lab in the building. So we were studying with bacteria, mostly E. coli, but the, so, so it's important to understand. I mean, if you are studying biology, I am like, talking to anybody out there. If you are studying biology, first things first. I mean, you have to understand the cell wall, the cell membrane, and what's happening with it. Just so there is this intermembrane space in between the outer membrane and the cell membrane. It's called periplasm. So that the whole thing surrounds the cell. The cell membrane also forms intracellular compartments in some bacteria to function to maximize the amount of membrane available for use by membrane-bound enzymes. Actually, that's the whole thing. Membrane-bound enzymes, because bacteria is such a small, organism. They are running out of space. I mean, it's just like small, tiny. So a lot of the enzymes are embedded in the membrane. They are built it. Imagine our homes. I mean, we have lots of stuff embedded in our walls, and like the walls and windows and the doors, and so think about like that. Think about your home, your house, and think about the exterior, covering the exterior wall of your house. Is it stone? Is it like aluminum siding, vinyl siding? So think about those. The cell wall determines and supports cell shape. So this is the actually most important part of this talk. The cell wall determines and supports cell shape. So especially in microbiology, like we have like the most of the taxonomy is based on like bacterial shapes. Some of them are like crooked, and some of them are like baton and sticky, and some of them are like globular, and the cell wall kind of decides and determines the shape. The cell wall determines and supports cell shape. So that's very important. I mean, that's why we have to study this cell wall. But our goal is to destabilize the outer membrane and lyse the cell wall to form a spheroplast. So that's the aim of the experiment. Destabilize the outer membrane. So I wish, like I said, I had the drawing, but it's just that it's a double membrane. Gram-negative bacteria has a double membrane, just one outer membrane, one periplasm in between membranes, and an inner cellular membrane. And if you get rid of the outer membrane with the cell wall, you create, or you form a spheroplast. It is in the shape of a sphere, globular. That's why it's called spheroplast or protoplast. In some publications they call it protoplast, and some they call it spheroplast. But in case of bacteria, because it becomes round, that's why it's called spheroplast. The cells become round after the procedure. So let's go down. Spheroplasts are round cells, like I mentioned, that lack a cell wall and lyse in hypotonic solutions, because that's the purpose of the cell wall, to protect the bacteria. I mean, the bacteria lives in oceans and lakes, and especially lakes. It's a hypotonic solution. In a lake, you have to protect the cell content. I mean, everybody knows the osmosis. I mean, you have to protect the cell content. Otherwise, it'll just suck up all the water, and just and the cell will be damaged. You need to protect the cell content so it doesn't leak out and etc. In terms of solutions, hypertonic, like hypertonic solutions, meaning like saltier seawater, for example. You know, it, it's a hypertonic solution. It's full of salt, and 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 this is a challenge for many fish because you are swimming in a sea of salt. I mean, it's a hypertonic solution, and then in the lakes, if the imagine a lake filled with ice water. I mean, all these like mountain rivers and mountain springs, and all that ice melting and forming lakes. Like for example, the Devil's Lake in Baraboo, Wisconsin. It is mostly of snow, ice. So that creates a hypotonic solution. To protect the bacteria, you need the cell wall. So in a spheroplast, there are none. Periplasm contents leak out to the solution after lysis of cell wall and the stabilization of outer membrane with EDTA reagent, causing periplasmic proteins entering the solution. You see my English kind of. I had some older. I had some the typos. Causing periplasmic proteins entering the solution. So this periplasmic space has proteins in it, enzymes, and maybe next week I will show another homework of mine regarding these membrane proteins and their like periplasmic parts and domains. So there is this place, periplasm. Many of us don't know very well. So periplasmic proteins. So when you are making periplasm, when you are making a spiroplast, and then basically you are getting rid of the periplasm because you're reducing from two membrane into one membrane. Our aim is also to perform alkaline phosphatase for a enzyme assay found in the periplasm of Escherichia coli. E. coli. So the periplasm of E. coli has this enzyme called alkaline phosphatase, and we will try to see if we can work with this enzyme, because once you remove the outer membrane and expose the periplasm, this enzyme leaks out into the solution, and then now you have an enzyme, and then you can do experiment with it if your periplasm, if your spiroplast formation is successful. Hydrolytic activity of alkaline phosphatase degrades substances such as colorless XP substrate. So so this enzyme has a hydrolytic activity, kind of it's like a digestion activity, So it works on this XP substrate. So this reaction results with a blue color product and it liberates phosphate from substrate. So that's what the enzyme does. So let's continue. And the enzymatic rate can be measured semi-quantitatively by analyzing color. See, most of the experiments, we are using color to understand the results. The enzymatic rate can be measured semi-quantitatively by analyzing color change of the sporoplast culture, given that the substrate is saturated. Yeah, I mean, as long as you have enough substrate, like if you don't put the color material, I mean, it won't change color. Not because there is no enzyme, but you don't have enough stuff. You don't have enough dye, to give you the color. So in these reactions, so the amount of materials and the amount of cells is always important. Just so it's in most of the experiments you have to measure the amount of cells. You have to measure the amount of material dye substrate, and same thing for human biology. I mean, we go to doctors and you have a low, like white the blood cell count. What happens? Well, you don't have red blood cells. You become anemic. You know, it's all it depends on the number of cells. I mean, we need white blood cells. We need red blood cells. We need platelets and thrombocytes and and, uh, and the leukocytes are like the whole the different set of cells. The just general name is leukocytes. So just keep that in mind. I mean, so we are doing all these experiments and keeping in mind that we had to control the numbers of these cells. Like any experiment, you have to know your numbers. Materials and methods. So culture, 3-hour culture of Escherichia coli. This is the strain, was inoculated from a 5 ml culture to grow young cells. 150 ml culture was harvested at 5,000 rotation per minute for 20 minutes in a JA10 rotor in high-speed centrifuge. That is the standard, actually. Centrifuge speed to harvest cells, 5,000 RPM. It works for yeast cells as well, so when you are washing cells and etc. or changing media, that's what you do. You pellet the cells and like dump the media and then put in some other media, etc. Especially when we were doing Western blotting experiments, first you have to have media with glucose and then reduce glucose, etc. or high nitrogen, low nitrogen, etc. So this is the buffer for the 40 ml 10 millimolar tris hydrochloric pH 8 was used to wash cells by centrifuging at 15,000 RPM. 15,000 RPM for 15 minutes in JI2 rotor. So this is a little bit faster one for 15 minutes. So see, that is, I guess, that usually this speed. See, it's actually unusual. The rate. Hope I didn't make a mistake there, but looks like real. So let's see. Let's see what happens with that. Actually, I had the material paper open, but that could be the true, because let's see. So that's the final centrifuge at 15,000 RPM for 15 minutes in J20 rotor. And wet weight of the pellet was determined to resuspend cells in 20% sucrose in 10 millimolar tris, 15 ml sucrose, gram wet weight. Maybe that is necessary to get rid of the the cell wall content and etc. And now, because you have to resuspend the cells in 20% sucrose, because now remember the cell wall is gone by now, in terms of the material, and now you have to have excuse me, you have to have some protective solution, 20% sucrose, not hypertonic, not hypotonic, like physiologic, serum, so that cells are not damaged. So 15 ml sucrose, gram wet weight. So that, we just measured the wet weight of the cells, and then you apply this amount of sucrose, 20% sucrose, uh, to those cells so that they don't burst open. I mean, that's the idea. I mean, to just serum physiologic. I mean, they're just preparing this. I think what I heard, like in nature, the coconut, the coconut juice, the stuff inside the coconut is like they say acts like a serum, and it's sterile, so in some cases people used it or not, maybe just something. So that's maybe it has 20% sucrose to so lysozyme 1 to 10 volume, and this sodium EDTA, that's chelating agent, kind of binds the metals, was added to the cell suspension and incubated 20 minutes at room temperature with stirring. Removed specimen. Actually, this is the enzyme that strips the cell wall, the peptidoglycan material, that polysaccharide material that surrounds it, the lysozyme. That's the enzyme. Lysozyme and the sodium EDTA was added to the cell suspension and incubated 20 minutes, and it does the job. At room temperature with stirring, removed specimen approximately 2 ml with Pasteur pipette. Final 10 ml culture was incubated for 10 minutes at room temperature with stirring, and approximately 2 ml specimen was removed with Pasteur Pasteur pipette. So that's there could be other protocols, like how to prepare protoplast for different cells. This is for a gram-negative bacteria, and and before I continue with the rest of the the experiment, it is important because most of the plasmid projects, all the genetic projects, research involves all these plasmids, and then it's not easy to get the plasmids inside the cells, especially for mushrooms, because now we have all these mushroom species producing all these anti-cancer compounds and etc., and you want to generate a drug or a medicine out of it. You have to study the biochemistry inside those cells, and to do that you need to carry out transformation, and meaning you have to get the plasmids inside the cells, and oftentimes when you strip the cell wall, the cells get damaged. So it is a very fragile process. Making a protoplast, making a spheroplast is very fragile, very sensitive experiment. And if you cannot maintain the cellular homeostasis, if you cannot maintain the cellular health during this experiment, you cannot get any DNA inside the cell to continue with the rest of the experiments. You have to be able to get a piece of DNA fragment inside the cell. So oftentimes, in case of mushrooms, I'm reading papers, and then they can now make a protoplast in, for example, king oyster, but it doesn't work for oyster mushroom, regular Pleurotus ostreatus. They cannot get a protoplast to carry the transformation with the plasmids. So they have to use a bacteria to do the transformation, like they do with plants. But so it is an important experiment, and yet today, this is like year 2007, and we are here now, 2024. Still, it's a challenge to prepare a healthy protoplast or spheroplast in the case of bacteria or E. coli. So the material methodology, all the numbers you see here, you can play with that. I mean, that's why I am reading this and that, and don't be so bothered with that. You can always change the numbers here and optimize it for your own lab, especially for preparing a protoplast. The general protocol gives you an idea, but what you have in your lab can be different, can be unique. So it's always important. What's really happening with these protocols, and what can you change and what can you not. So, by the way, let me get some tea. Normally, like making these talks, you have to have coffee. It helps. If you don't have brain connection. Normally, I don't do my talks without coffee, but at this stage now, I'm okay with coffee, and just it's enough. So spheroplasts were observed under the light microscope. Alkaline phosphatase activity was detected after 30-minute incubation of 240 and 480, twice as much microliter cultures with respectively 240 microliter of substrate, this XP substrate at 37 Celsius degree. So because if this protoplast formation was successful, then you would have alkaline in the solution. That's this enzyme, alkaline phosphatase, and then you can measure its activity with this substrate, which gives the color. So let's go on. EDTA and lysozyme caused spheroplast formation in E. coli. Reagents destabilized the outer membrane and lysed peptidoglycan cell wall, which eventually resulted in spheroplast in isotonic sucrose 3 solution. See, that's I forgot the name of the solution, so that's why I'm reading my reports again and again. It's been some years, and it's isotonic solution, hypertonic, hypotonic, and isotonic, which is like serum physiologic. It is important to prepare this isotonic sucrose solution for mushroom researchers as well. I mean, because if you are damaging, the cell during this process, it will not help with the transformation or with whatever you're doing, because this is just the start of the experiment. Periplasm protein alkaline phosphatases was also detected with semi-quantitative assay, meaning just change of color. Color change suggested that substrate was limiting factor, unlike experiment was planned. Successful spheroblast formation yielded abundant enzyme, and 400 microliter substrate culture mixture showed less coloring than 240 microliter, proving that substrate was a limiting factor in this experiment. So there was abundant amount of enzyme, and then which successfully turned color of this amount of material, 230 to 240, and then this 400 microliter substrate, I guess, was a bit lighter in color. So that just shows the the rate of the material or the amount of enzyme. So proving that substrate was a limiting factor in this experiment. Untreated cells displayed slight coloring resulting from cell wall damage disruption. See, remember that maybe during the centrifuging or all these experimenting and pipetting and etc. So maybe there was a minor leak, and then that just saw that untreated cells, the cells with the cell walls, they still have a very slight color formation due to the cell wall damage and disruption. Imagine, like, this is healthy cells and not treated with enzyme and etc. EDTA or lysozyme, but still gets disrupted during the experimental procedures. And so that is to keep in mind. Like, you're trying to get a healthy protoplast, you have to be very gentle with the cells. I mean, like, I remember even for transformation, I remember my professor telling me, like, be gentle. I have used the ml pipetter tip, and then, like, gently to just swirl and mix the cells, and because you're trying to get the DNA inside the cell, and you should not damage anything. All this experimental procedures, even with the cell wall, they can damage. Periplasm leakage during the process, like pipetting. Okay, so it's important. Like, every step of these protocols are important. You have to be gentle. You have to be very careful. Fragile. Life is fragile. That is to keep in mind. That is the final idea of this experiment. Life is fragile. It needs protection. It needs cell wall. So that is the kind of idea. And that's and let's see the results. Okay. See, we measured the wet weight of the cells. This is the weight, and then times 15 ml. I think sucrose, 20% sucrose. That was the ratio. So wet weight, and that's the color change. So that is. Let me see. Just go up. This is regular cells. See, because of the leakage, maybe pipetting error, and then some cells changed color, turned slightly blue because of the pipetting error and etc. But with this amount, it didn't create any dye. And when it comes to spheroplasts, 240 microliters was blue, and this was lighter blue. This was blue, and this was lighter blue. And this, in this case, again, blue, and then lighter blue. And same thing. So different different groups. So that's the idea, pretty much. That's the idea. So alkaline phosphatase activity of spheroplast suspension. Like, well, once you get rid of the outer membrane, this enzyme leaks into the solution, and then as you see in this spheroplast, you see the color formation because of the enzyme activity on this material. So if you increase the amount of the dye, amount of substrate, you get better colors. I guess you see just the a little bit darker blue, I guess. Still, compared to this lesser tone of blue, but so this was so the concentrations are important, like in all these experiments. I mean, that's just whether you're just working with cells or enzymes, or even if you are like fermenting yogurt, you have to have enough yogurt, like a case for a 5 liter, and you have to have enough material. Usually, that's what we do. For I think 4 liters of milk, boiled milk, you need kind of at least half a cup of yogurt, good yogurt to start the fermentation, good fermentation, which takes about 3-4 hours. So it's all about cells. It's just so that is the idea. Wet weight of the pelleted culture and reaction volume was used to estimate reaction rate. However, dilution of XP did not increase rate since the amount of enzyme was abundant. It's talking about this as just see that if you put the dye twice as much, that is like 100 microliter of substrate and 200, and then in this reactions that's called limiting factor. It's talking about say dilution of XP did not increase rate since the amount of enzyme was abundant. So it was so it's a matter of ratios, enzyme ratio, enzyme substrate ratio. That tells you that pretty much the numbers are here, same amount of cells, compared to one another, and even if you double the concentration or amount of dye, you still get the same amount of color. The amount of enzyme was abundant, so I guess that was that was dark as it gets, the darkest maybe, that I guess. Okay, and this is the shape. This is regular cells, 200 microliter, 400 microliter. They have this, like a kind of elongated morphology, and then spheroplasts, and they that's why they're called spheroplasts, kind of round shape. They lose their shape, like the bacteria have different shapes. you know. So these are round because now they don't have the cell wall pegged to the peptidoglycan structure. You know, it's just cell membrane. Spheroplast formation of EDTA and lysosome treated cells. So that is the kind of end of experiment. So that's the discussion. Reaction rate was one of the goal of this study, and limited substrate did not allow enzyme-dependent reaction rate to be determined. I said. that's what she said. That's what I said. So that's what I wrote. I guess reaction rate was one of the goal of this study. Like I said, in these reactions, in enzymatic reactions, it's always the important, how much enzyme and you have and the amount of substrate and etc. Cell culture density played an important role in estimating enzyme amount. Higher the number of cells and higher the concentration of enzyme, you would think and you would hypothesize. So spheroplast formation was successful and yielded abundant alkaline phosphatase for assay. So that was a successful experiment. We have used fresh 3-hour culture to be able to see formation. This is fresh culture because in time they can die. That's another with these cultures. I mean they always, because they go through mitosis in a culture, and like sometimes we would leave them on the shaker at night and then come in the morning and then fresh the cultures, refresh the cultures all the time, because you cannot use the overnight culture. You can start there. You can use them because they have live cells, but most of them can be dead. So that is the kind of rate, 3-hour culture, fresh 3-hour culture, to be able to see formation. Substrate we have used could have been substituted with another. You can use other materials, I guess. This would allow determine more realistic enzyme reaction rate, maybe more sensitive substrate. So that is to say, like, you can always play with these experiments and see if you can get a sensitive coloration rate or not. So designing experimentation is important. I mean, it's part of the biology. It's not always like learning and and memorizing and doing this and that. But most of the time, biology is setting up the right experiment. It is the art of experimentation. Biology is the art of experimentation. That's why the name of this class is experimental microbiology. You have to be able to establish a successful experiment, with the successful protocol, with the successful rates and the timing and etc., with the material and the concentrations. So it's a hands-on science. Biology is a hands-on science. Same formation could be observed in different gram-negative cells. See, this is E. coli, and maybe you want to try the same protocol or same experiment with other gram-negative cells. Cell wall and outer membrane play important roles in terms of antibiotic resistance. That's how it started, actually, this antibiotic resistance, because it has the enzymes. In this case it was alkaline phosphatase, but most of the time this resistance enzymes are there too. plays cell wall and outer membrane play important roles in terms of antibiotic resistance, hydrolysis of nutrients, etc. Very critical part of the cell, this outer membrane and periplasm. It's like a garden for the cell. They think about it. That this cell wall and the periplasm is like the garden of the cell. Think about it. it's so a lot of things happen in it. So antibiotic resistance and guess what? Gram positive bacteria, they don't have the antibiotic resistance. So most of the time you find gram positive bacteria in extreme places. Where regular gram negative cells cannot live. There is a geographic difference. For example, in our guts we have gram positive bacteria, not gram negative ones, because because of the antibiotic resistance. The soil is full of these genes. The soil is full of antibiotic resistance genes. I'm talking about soil, and then that's why it's a problem. Every 10 years we have to develop or generate a new antibiotic. Why? Because the the soil is the reservoir, for these genes. Different bacteria, all gram-negative bacteria, they have all these enzymes, in their periplasm and in their outer membranes, giving them antibiotic resistance. Because why? Because most of the fungi generating all these antibiotics living in the soil. So they, both they have to protect the fungi. Needs to protect its food source, and the bacteria needs to protect themselves from the fungal antibiotics. So it's important. Like I said, gram-positive bacteria, you find them in mines and in acidic solutions and in halophilic soils and waters, and all the extreme places you find gram-positive bacteria because they don't have the antibiotic resistance system in their membranes, in the outer membrane and periplasm, and there are many theories regarding gram-positive bacteria, and maybe that's how life started, because that we still don't know how the eukaryotic cell formed. Maybe some gram-positive bacteria got inside the gram-negative bacteria. It's something like that, because it's still weird, that these we still don't know how we got the eukaryotic cell, which has mitochondria in it, which is like a bacteria, and which has chloroplast in it, which is like another, which is like a bacteria as well. So it's important, this cell wall stuff, and then the gram-positive, gram-negative bacteria and where they live, important. They also determine the shape of cell, which in fact related to overall niche of the species. I use the word niche, like I said, because all these bacteria living in soil, living in our guts, living on the skin. It depends on their cellular structures. Whether they are gram-positive, gram-negative, and gram-negative ones are kind of winning the game in the environment, but still there are many gram-positive cells living in like weird places, in mines and in thermal vents and etc. I think that is the end of the report. So that is important. when you like Google a cell a picture on internet, you just get a shape, round shape, and that's what it shows. But in in fact, in yeast cells, uh, they're like round, and budding yeast is like a potato, little potato buds growing. They're kind of a potato bud yeast cells. That's why it's called budding yeast. Under microscope, you always see, little round structures, new baby cells growing out of mother cells and etc. In mushrooms, the spores, they're mostly cylindric shape, rod shape, like 1 micron times like 10 micron, 11 micron, and you can see the shape of these various mushroom species under the microscope. And oftentimes the mushroom definitions are based on spore colors and spore shapes. So if you are a mushroom hunter, and if you wanna like learn more about mushrooms, try to get a microscope. I suggest, if you can find for a decent price, maybe $1,000 or 500, you can get a second hand. But if you are a dedicated mushroom, enthusiast, you are if you are into mushrooms, then maybe you can look at their spores and do identification, do better identification, and even make better cultures. You can, if you can isolate the spores. There are some videos on the internet, with the you can check. There are methods for like if you see the spores in the solution, you can sometimes suck with the like glass rods and etc. And then then pick, because they have a certain shape. That's the whole idea. Every mushroom spore has a specific shape. Every bacterial cell has a specific shape, and you can maybe you can use dyes, like dyes that are not harmful, so you can dye the cells, and then you can maybe pick them easier. Just to give you an idea, that's the whole idea. I mean, the gram positive, the name comes from the stain, the gram stain. So it's important, and many of the mushroom spores have colors and coloration, and let me tell you one more thing. Because now I am also investigating macrophagy, the insects, for example, eating mushrooms, the slugs eating mushrooms, and then the cell wall of the mushroom spores sometimes play an important role in the diaspora of these spores, because if the spores can withstand the stomach acid and the digestion of these animals, they can be carried to new places with these animals and insects. And then many insects carry these spores, like bees carrying pollens. Compare bees carry pollens, butterfly carry pollens, and etc. The pollination process, same thing applies to mushrooms. And because it is understudied, we have no idea, because some slugs eat mushrooms, and then they get contaminated with the spores. They carry the spores, the slugs and insects and all these ground insects, ground beetles, flying beetles, those deer-shaped beetles. That sometimes they like they have Lucanus, I think, the name, the genus name, Lucanus cervus, like the deer-shaped, the deer beetle. They sometimes fight. That on the internet you will find. I mean, they are ground beetles. They live among the leaves. I've seen it in Sweden, in the Stockholm Museum of Natural History. They had this Lucanus species in a in a in a glass terrarium or something, and then they showed us, those they said we have a giant here, we have the monster, in so it is important, yeah, so the shape and the function of these cell walls, of the cells are important. In mushrooms they are important, in bacteria they are important, in humans they're still important, because we get dehydrated. Drink water, drink water. By the way, we cannot stay healthy without water. And even same thing for us too. If we drink drinking regular water, not too acidic, like fountain water, which has some level of calcium in it. If you drink water with like the pure water, that also can help with your headache. So, drink water, isotonic, I should say. Of course we cannot drink sea water, but drinking pure water is not so good as well. That's what I hear. So it's good to have mineral water. see, I love it. Try drinking mineral water. So it has magnesium in it too. Everybody's talking about magnesium. Stuff has magnesium too. And it doesn't have sugar. I mean, trust me. You'll get used to it. When you first try the real mineral water. It may be bitter, but once you get to it, you will love it. In Turkey, it's just in summertime, mineral water. Okay, so that is the end of the talk, ladies and gentlemen. Again, I would like to mention I'd like to mention the earthquake victims that happened a year ago, February 6th, and thousands of people died. It was a horrible earthquake, 7.4, and three of them happened, one after another. So it was just a disaster. It was a disaster, and today is the anniversary, and then we are honoring all those who lost their lives under the rubble. It was just painful. I'm sorry, really. It was just some of them waited for help. It was so hard. All right, thank you very much. Please subscribe to my channel, and I will try to prepare more talks. Thank you very much. Have a good night.